everyone. This is Angie Draculich, Editorial Director of BioPharm International. I'm here today with Dr. Jayne Telford, Bioanalytical Training Lead at NIBRT. NIBRT is the National Institute for Bioprocess Research and Training. The organization provides training, educational, and research solutions for international bioprocessing industry and state-of-the-art facilities. Located in South Dublin, it is based on an innovative collaboration between University College Dublin, Trinity College Dublin, Dublin City University, and the Institute of Technology Sligo. Today we're here to talk about product characterization. Thank you, Dr. Telford, for joining me.
Thanks, Angie.
So the primary goal of product characterization is to ensure a product's safety, purity, identity, and potency. And ICH-Q6B specifically calls for determination of physicochemical properties, biological activity, immunochemical properties, purity, and impurities to establish specs. Can you go into more detail on why these characteristics are crucial for overall product and patient safety?
Sure. So we're talking about protein-based biotherapeutic drugs here. Therapeutic proteins elicit the desired effect in a body or in a patient by interacting with receptor proteins, with other proteins, or with other targets to enhance or inhibit signaling or interactions in the body. In order for these proteins to have their desired effect, they must have the correct sequence, the correct size, the correct structure, and the correct post-translational modifications so that they are recognized by their binding partner. And also they must be biologically active and have the correct physiochemical and immunochemical properties to elicit the correct response. These therapeutic proteins are generally produced by cultured cells in large bioreactors, and they're harvested and purified. But the actual generation of the proteins in the cell occurs along a complex pathway that involves transcription of the DNA in the nucleus and translation of the protein in the cytoplasm. The initial polypeptide chain is further processed by folding and by post-translational modifications, producing the final biologically active protein. These processes are very complex and therefore errors can occur. So for this reason, during the production of a therapeutic protein, it's vital to monitor all aspects of the protein to ensure that its sequence, its structure, purity, and its stability are correct and consistent. Errors to the sequence or the structure of the protein could result in the drug not having the desired beneficial effect in the patient. Or even worse, it could affect the safety of the drug and could have the potential to cause detrimental effects in the patient, such as eliciting an immunogenic response, which could lead to anaphylaxis, and that's obviously very dangerous. Basically, all proteins then need to be analyzed in detail to ensure an active, pure, stable, and safe drug can be administered to patients. To give an example of an error occur in a therapeutic protein, it could be the presence of non-human glycosylation, for example, alpha-gal, on the surface of a protein. This will be recognized by the body as being foreign, and an immune response will be elicited to get rid of the drug. This incorrect product would not be active in the patient and could actually endanger the health of the patient. So obviously we want to avoid this from happening.
Right, thank you so much. When should that type of testing be performed?
So some people might think it's sufficient to only characterize the product during the discovery phase and then again at the final product stage. However, this is not the case. It's advised by the FDA and the EMA that the product is analyzed at all stages during the processing pathway. So during discovery, upstream processing, downstream processing, and right through to the final product. This is to ensure that the product is consistent, active, stable, safe, and pure, right through from the start to the finish. Analysis at all steps also allows any changes to the protein or other problems, such as contamination, to be detected immediately, thereby allowing easier determination of where the problems actually are occurring and enabling faster investigations to be performed. If analysis is only carried out at the start and at the end, it would be very difficult to pinpoint the source of the problem, and lengthy analysis would need to be carried out to rectify the issue.
Great. Now, I know that there are many regulations the industry must follow. I was wondering if you could briefly discuss the key United States and the European Union regulations that describe requirements for protein characterization of biologics and what those organizations are looking for today.
Yeah, so as you say, there are many different regulations. So numerous US and EU regulations exist for biologics, and they're categorized for different types of proteins, for different types of production pathways. So some examples, there are guidelines for recombinant proteins, there are guidelines for monoclonal antibodies, and there are guidelines for biosimilars from the various regulators around the world. However, the guidelines for all therapeutic proteins remain similar throughout the different categories, and reference to the ICH quality guidelines provides a good basis for understanding what is required, as this represents harmonized guidelines based on the various regulations. And so just as an example, I'll detail the characterization requirements for monoclonal antibodies. So information must be provided on the identity of the monoclonal antibody, so for example, its sequence of amino acids, on its structure and size, on its potency, on its heterogeneity, its glycosylation, immunological properties, its specificity, cross-reactivity, if there's any aggregation of the product, and also process-related impurities. So extensive analysis would be required to provide this information on a monoclonal antibody, but the similar requirements are outlined for other types of proteins and processing types.
Okay. And when performing these analyses, what techniques and instruments are most commonly used today?
Yes, there are many protein characterization methods commonly used, all at differing levels of complexities. The simplest methods would include protein assays, SDS page, isoelectric focusing, Western blotting, and ELISAs. Protein assays such as the Bradford assay are simple methods that are used to determine the protein concentration of the sample. SDS page or isoelectric focusing are frequently used to separate the components of a sample and to gain information on its identity and purity. Western blotting and ELISAs are routinely used to identify and confirm the presence or absence of the protein. Another commonly used method is capillary electrophoresis. So this is very useful for gaining information on the size, charge, purity, and identity of proteins. And it provides similar information to SDS page and IEF, but it's much easier to use, it's more reliable, and more quantifiable. There are other more complex methods, such as HPLC and mass spectrometry. These are extremely valuable. Reverse phase chromatography, size exclusion chromatography, and ion exchange chromatography are all particularly useful for separating the components in a protein sample and for providing information on the identity and the purity. And mass spectrometry and peptide mapping are useful for confirming the amino acid sequence of the protein and for identifying the protein. So this is just an overview of the commonly used methods, but obviously there are many other useful ones that are widely available and widely used.
Right, and I know as the bioanalytical training lead, you must be following this area very closely. I was wondering if you've seen any new techniques emerging or if you've come across any gaps in technology in your own work that maybe you're hoping will be addressed soon.
Yeah, so of course there are two in particular that we're kind of talking about at the moment. So some of the emerging techniques involve the analysis of post-translational modifications of the therapeutic proteins, and this can be very difficult. And so some examples of post-translational modifications are glycosylation, phosphorylation, and oxidation. And of these, glycosylation is the most common post-translational modification. It can be very complex and diverse, making the analysis very complicated. But advanced glycoanalytical technologies, including HPLC, UHPLC, mass spectrometry, and capillary electrophoresis are emerging, and this is enabling companies to gain increasing volumes of information on glycosylation. However, this area will benefit from increased advances in technology, especially in the analysis of O-linked glycans, which are much more difficult to analyze than N-linked glycans. Okay. There's another area of interest as well, so it's protein aggregation analysis. Proteins can aggregate under certain conditions, and this would be formation of dimers, trimers, and larger multimers. So they can affect the activity, the immunogenicity, and the stability of a therapeutic protein. The detection of the aggregates is very complex, but a lot of techniques again are emerging to help with this. So such as dynamic and multi-angle light scattering, analytical ultracentrifugation, and size exclusion chromatography, just to name a few.
Great, thank you. I know that validation is always important to the industry, and I was wondering where qualification and validation of these analytical methods come into play when you're dealing with product characterization.
Yes, of course. So qualification and validation are indeed very important. All laboratory equipment first needs to be install qualified, so IQ'd, and operationally qualified, OQ'd, prior to validating the actual test methodologies. And the validation of analytical methods is covered well in ICH Q2, which is entitled Analytical Validation. A significant part of this validation involves ensuring that the software is in use by the equipment meets the requirements also. So the requirements of Title 21 CF4 Part 11, which is entitled Electronic Records, Electronic Signatures. And then the IQ, OQ, and PQ protocols and reports need to be prepared and approved for each piece of equipment and for each test methodology before analysis of the product can begin. So once this has been achieved, the tests can then be used to characterize the product throughout the manufacturing process.
Okay, you've given us quite a detailed overview of product characterization. Could you talk a little bit about some of the challenges the industry faces when they're doing these tests?
Sure, yeah. One key challenge would be for companies to locate skilled scientists. So the advanced methods of protein characterization can require specialized training for scientists to be able to analyze the product adequately, and to allow them to troubleshoot when problems arise and spot method-related errors as they occur. Another challenge is that the biologic protein product characterization typically requires offline sampling, and this can take time and would benefit greatly from advances in online technologies. This would facilitate real-time analysis of the product as it passes through the manufacturing process. So, for example, a sample could be analyzed as it directly comes from bioreactor. One other major challenge, of course, is in the detection and prevention of contamination. So this would really be helped by the development of rapid analytical methods for viral and microbial contaminants at each stage of the processing.
And what about any best practices? Can you offer any for listeners?
Well, one thing that we can help with at NIBRT is in the training of the staff. So with helping with the skilled scientists, it's important that these analysts understand how the product characterization methods work, why they're performing them, how to analyze the data, and when things go wrong, that they can spot the problems and troubleshoot where necessary. So at NIBRT, we provide numerous training programs on protein characterization methods, protein aggregation detection, glycan analysis techniques, and QC methods, along with many other training programs such as those on upstream processing, downstream processing, and fill finish. And they are all with very hands-on, practical manner. So they're designed to support the bioprocessing industry.
Great. Well, thank you so much for your time today. I greatly appreciate your insight and expertise on product characterization.
Thanks, Angie.